Hello, this is the histology tutorial on bone presented by the SchrDbb with reference Histology, Atlas of Histology and Functional Correlation by Dongmei Cui. So, here we're going to continue with bones. So we start first with the introduction of bones. That the bone is constitutes a part of the body which is a supportive connective tissue. So it consisting of hard mineralized extracellular matrix and all that. The next point is that we need to know that since a bone is just a connective tissue, we are going to divide the bone in cells of bones, matrix of bones, and fibers of bones, because a bone is made up of an organic and inorganic part. The organic part of the bone can be considered as the fiber of the bone. The inorganic part of the bone can be considered the organic part of the fibers of the bone. Inorganic part of the bone can be considered as the matrix of the bone, and then the cells of the bones is the third part. So the matrix of the bone, you need to know that the matrix of the bone is made up of organic matrix. So if you don't want to consider the fibers of the bone as being the matrix of the bone, or you want to consider as being the matrix of the bone, just call it as organic matrix. It's mostly made up of type 1 collagen fibers. And now the ground substance of the bone is made up of the chondroitin sulfate. So it's made of the ground substance of the bone is made up of chondroitin sulfate or keratin sulfate. The next one is the inorganic part of the bone. The inorganic part of the bone, inorganic matrix of the bone, is made up of hydroxyapatite. Now the next thing is bone cells. So the bone cells can be made up. There are three main bone cells. The first one is the osteoprogenitor cells. The second one is the osteoblast, and the third one is the osteocyte. We also have the fourth one, which is osteoclast, but osteoclast is mostly considered as a wandering cell because it started from the bone marrow and then entered into the bone. While the osteogenic cells, the osteoblast, the osteoclast were all fixed cells. They were produced in the bone and then they live in the bone. Now the types of bones. So what are the types of bones? Types of bones can be classified. So if the bones can be classified now. You can have primary and secondary bones. That's the first division. Primary and secondary bones. Now primary bones are just called immature bones or woven bones. Those bones are mostly present when you are in a fetus, as a fetus. So when someone is a fetus, that one is going to be immature woven bone. Now secondary bone is a mature or lamellar bone, also called mature lamellar bone. Now you also have division of bones as being long bone, short bone, flat bone, or irregular bone based on the shape. But based on the maturity, we have primary and secondary bones as a main type of bones. Now secondary bones, which is also called mature lamellar bones. Lamellar because it's made up of lamellae, and primary bone is immature woven bones because it's made up of woven like aspect. Now the mature bones can be divided into two. We have compact bone and cancellous bone. The compact bone is also called cortical bone. Compact bone is also called cortical bone, while cancellous bone is also called spongy bones. Now for bone development, you need to know that there are two main types of bone development. You have intramembranous ossification, when you have ossification from hyaline cartilage that was present inside the fetal skeleton, and you have intramembranous ossification, when there is ossification from membranes, like production of the frontal bone, temporal bone, that's intramembranous ossification. And then the next one is endochondral ossification. Endochondral ossification now is produced from hyaline cartilage, when there is ossification of hyaline cartilage in the fetal skeleton. So now this is the diagrammatic morphology of a bone. We need to know that this a mature bone is made up of two main parts. The part which is located laterally is a compact or the cortical bone, while the part which is located centrally is the spongy bone, or it's also called the spongy bone or the cancellous bone. Now we need to know that the compact bone is made up of osteon. So as you can see, you see osteon here. So the osteon is made up of circular lamellae, and then the inner part of the osteon is made up of the Harvesian canal, as you can see here. Now connecting to osteon, you can have now the Volkmann canal with the blood vessels and the nerve fibers. Now we have the space of the cortical bone and the cancellous bone. Since they are also called lamellar bones, they are containing lamellae, as you can see here. So we have inner circumferential lamellae, as you can see here. We have outer circumferential lamella, we have interstitial lamellae, we have the osteon here, we have the Harvesian canal, we have the Volkmann canal. This is this cancellous bone here, and the cancellous bone is made up of trabeculae. The trabeculae are these rough endings, as you can see here. Now let's see the types of bones now. So let's visualize the types of bones. So the types of bones, you have compact bones. So compact bone, this is the histology section of the compact bone. So on the histology section of the compact bone, you have concentric lamellae, as you can see here. We have these are Harvesian canal. This is a particular osteon which is made up of concentric lamellae. As you can see, we have the Harvesian canal inside. Now connecting the two Harvesian canal at the Volkmann canal that you can visualize here, depending on the section. You may not visualize the Volkmann canal when you're in cross section, but usually in histology the section may not just be a cross section, but it can be an oblique section. So that's why you can both visualize the Harvesian canal and the Volkmann canal in the same plane. So this is the lacuna. So here where the dark structures which are located here is the lacuna, and the lacuna is a space that contain a particular osteocytes. So those are lacuna here, lacuna, and they contain osteocytes inside. And these are interstitial lamellae. Interstitial lamellae is between two osteon. The next part now is a compact bone. So since this is another part of the compact bone, here you can also visualize the interstitial lamellae. We can say we can visualize the Harvesian canal. You can visualize the constrictive lamellae, concentric lamellae, and you can visualize the cement line. Now this is the diagrammatic representation of an osteon. The diagrammatic representation of an osteon show inside the Harvesian canal. Then we have concentric lamellae. We have canaliculi, which are located at each lamellae on each line of the concentric lamellae. So we have concentric lamellae. We have lacunae, sorry, not canaliculi. We have lacunae, which are located here on the lines of each lamellae. Is it clear? And then the next one is canaliculi. Canaliculi these are the lines which these are some canals which connect the two different lacunae. Lines which connect the different lacunae, canaliculi. And then the last one is the cement line. The cement line is a line which encloses a particular osteon. As you can visualize here, we have a cement line which shows that this is one osteon which is complete. Now in between here, now we have the lamellae, so it closes one osteon. So to close all this osteon, here is the cement line, and then the cement line is a bit whitish in color. It's not that dark in color when you use this thing. And the stain here that they use is the ground specimen. They are unstained. So there is no particular stain that is used in these two cases, a ground specimen. Now, the next thing now to visualize is going to be the compact bone and the cancellous bone. So the compact bone and cancellous bone, you need to know that our compact bone are much more on the long bones, are much more going to be at the periphery, while cancellous bone are much more going to be at the center. So this is the bone marrow here. So the cancellous bone are going to be at the center, and they mostly consist of trabecular. This is a trabeculae, and these are the epiphyseal plates here. This is the articular cartilage here. So continue with the next part. So here inside we have the periosteum. So at the level of before the compact, go to the compact bone, we have first the periosteum that overlies the bone. So the periosteum is made up of connective tissue. Dense, it's made up of connective tissue. Is it clear? So there's connective tissue. Now the next thing after so at the level of the osteum we have osteogenic cells, which is located at the level of the osteum. Now inside we have osteoblasts, which are located here, and far inside, when you're at the level of the compact bone, now we have osteocytes, which have different arrangement of the osteum. Now this is with the hematoxylin and the eosin stain, so that you can visualize this. And inside here we have endosteum. So now with the cancellous bone, so cancellous bone or spongy bone, which is located in with nasal decalcified bone. So they are also called nasal, or they are also called decalcified bone. So these are the spongy bones. They are mostly made up of trabeculae. Normally in compact bone you have osteon. So osteon, concentric representation of laminae, you have concentric laminae. While in spongy bone you instead have trabeculae. Is it clear? So you have bony trabeculae or species. As you can see here, this is the spongy bone. This species here are the trabeculae. So trabeculae are also called species. Is it clear? And they are closer to the bone marrow. So now the development of the bone. So the development of the bone is divided into two. We have intramembranous ossification, and then we also have endochondral ossification. Intravenous ossification is a process of bone formation involving the transformation of condensed mesenchymal tissue into bony tissue by different mesenchymal cells into osteoblasts and osteoids. So that is intramembranous ossification. So it is conversion for the bony process involving the transformation of a condensed mesenchymal tissue. A mesenchymal tissue was an initial connective tissue. Is it clear? You first have an initial connective tissue because we said among the connective tissue we had mesenchymal connective tissue, which was an embryonic connective tissue. When there's conversion from that mesenchymal connective tissue to produce a bone, it's a bony tissue, it's going to be called intramembranous ossification. Here what happens is that we first have osteoid. Osteoid is first produced an unmineralized new bone. Then the osteoblast now going to secrete now the calcium, and then the phosphate that is going to now mineralize the matrix. As you can see here, we have the level of the osteoblast. Here is when it is mineralized, when it's becoming very pinkish. You know that the cartilages are not mineralized. That's why the cartilage are pale pink. But the more you are pinkish, the more you are mineralized. So that's why at the level here is no cartilage. It's only connective tissue, mesenchymal tissue. The mesenchymal tissue, these are the mesenchymal tissue, and then the osteoblasts are located outer part of the mesenchymal tissue. The more the osteoblasts which are located at the outer part of the mesenchymal tissue secrete calcium and phosphate, the more the bone is going to increase in size. Is it clear? So we have osteo inside here, the bone matrix already. We already have called osteocyte. They at work. At the beginning, the first osteoblast starts in the center, and when they proliferate and they secrete the matrix, the one that started stay in the center to produce the osteocyte, while the one which are outside continue producing calcium and phosphate. They continue producing calcium and phosphate to continue mineralize the bone until the whole mesenchymal tissue, that's the membranous tissue, is going to be mineralized. Is it clear? That is it. So that is it for. So you have this bone here, bony matrix which is produced here is the osteoid. So at the first we have mesenchymal tissue. The bony matrix here is going to be osteoid. It's not going to be fully mineralized, but it's going to be partially mineralized only. So when it becomes fully mineralized now by the osteocyte, which are present at the center now, is now going to be the bone. So that's it. So that the next thing now you have this is the osteoblast shown on electromagnetic electron microscope. So here we have the cell. This is cell here, and the cell here has the nucleus with nucleomatin. You can see the type 1 collagen fibers here. We have the osteoid tissue that has been produced. This is the mineralized matrix. The osteoid tissue is produced. The mineralized matrix is because of the osteocyte inside. The osteoid tissue is produced by the osteoblasts. and it consists of the type 1 collagen. When it is completely mineralized, it is now by the osteocyte, and it's called the mineralized matrix. Now, endochondral ossification. So endochondral ossification is a process of formation of the bone from the hyaline cartilage. That's it. So it's located from, so when you want to convert from fetal skeleton to adult skeleton to mature skeleton, is going to occur by endochondral ossification. So, and also the ossification which occur in growth of the epiphyseal plate is also by endochondral ossification. So that's it. So yeah, that's what happened. Now we have the epiphyseal plate. You need to know that there are different zones of growth in the epiphyseal plate. The first zone of growth in the epiphyseal plate is the reserve zone. The second zone of growth is the proliferation zone. The third zone of growth is the hypertrophy zone. Then we have the reserve zone. The second is the proliferation zone. The third is the hypertrophy zone. The fourth is the calcification zone, and the fifth is the ossification zone. So those are the zones of epiphyseal plate. At the apicalmost part of the epiphyseal plate is the reserve zone. And now in the reserve zone, what happened there? In the reserve zone, the cartilage or the cartilage chondrocytes are inactive, and the individuals are not arranged in isogenous group. So there are cells here, but they are not in isogenous group. They are inactive. Is it clear? So you can just consider this part as being like the perichondrium. So the cells here are inactive. So you have cells here inactive, not arranged in isogenous group. Now the proliferation zone. The proliferation zone chondrocyte undergo frequent mitosis. Here there is frequent mitosis indicative. So you can also, when you see the stain, since there is frequent mitosis, you may also even see chromosomes. You may not see chromatin and heterochromatin in this case. You may see chromosomes actually, which are splitting chromosomes inside the cells with the hematoxylin and eosin stain. You may see chromosomes, not euchromatin and heterochromatin. So I think because there is high proliferation here, as you can see some dots here as if the chromosome are splitting into two directions. So because they are highly proliferating, there is high mitosis that occur here. It's indicative of interstitial growth of cartilage. The next is the hypertrophy zone. The hypertrophy zone, the chondrocytes become mature and their size increases markedly. So this is the hypertrophy zone where the chondrocytes are mature, the size increases like that. You have the size of the chondrocyte increases, they become mature. So the isogenous group are clearly evident in this hypertrophy zone. Now in the fourth part, we have the calcification zone. Calcification zone, we have cartilage matrix become calcified. So the matrix of the cartilage will start because it become calcified. That's why it leaves from pale pink, it becomes more pinkish at the calcification zone, become calcified because and the chondrocyte died because nutrient and oxygen, so there is no nutrient since the chondrocyte here are very far, because it said that the inside here there is no blood vessels. Since the chondrocyte here are very far from the blood supply at the level of the perichondrium, because that you have to consider this reserve zone, the uppermost part as the perichondrium. Since it's very far, the cells here don't have oxygen and blood supply, so they are going to die. Is it clear? So when they die, they are going to live now place now to another type of cells, and that other type of cell now are the osteoblasts. Is it clear? Now the osteoblasts now are going to be produced now. The osteogenic progenitor cell and the osteoblasts are going to be produced, which are now going to produce the ossification zone. They are completely going to produce the normal bone matrix. Now this is the representation, the diagrammatic representation of the development of the long bone. It starts with the cartilaginous model. So we have the cartilage of the fetus, then the development of the cartilage, then you have the bone collar. Then when you have, so since inside here they are going to die, you have bone collar. We have now primary ossification when there is blood supply here. the osteoblasts now are going to proliferate here after the blood supply, they proliferate, and then they start producing the osteoid. Now when the osteoid is produced, you have secondary ossification area. As you can see here, when you have secondary ossification area, you also have proliferation of osteoid. And now after the proliferation of osteoid that's produced by the osteoblast, now the osteocytes now are going to now fully mineralize now that osteoid to produce now the bone matrix. Now osteosarcoma, what is osteosarcoma? Osteosarcoma is just referred, it's also called osteogenic sarcoma. It is just a malignancy of the bones. So when it's a primary bone malignancy. So when you have a malignancy of the primary bone malignancy, which is due to the bone itself, the bone parenchyma cells, it is osteosarcoma. How do you visualize osteosarcoma may be osteocytoma, that is when it's due to excessive proliferation of the osteocyte. It may be osteoblastoma, that is due to excessive proliferation of the osteoblast. It may be due to osteoclastoma when it's due to excessive it may be due to osteoclastoma when it's due to excessive proliferation of the osteoclasts and all that. Is it clear? So when you have all this excessive proliferation, you can think of that. Now the next thing is you may have the tumor cells. So on the histologic view, you may have tumor cells, you may have the osteoids. So you have tumors, you have excessive proliferation, and then there is no particular pattern of growth here. The next thing is bone remodeling, NASA. So in bone remodeling, the main thing that are involved in bone remodeling is osteoclasts and osteoblasts. So involved in bone remodeling, and osteoclast is influenced by the hormone calcitonin. So it's under the influence by the hormone calcitonin, which is secreted by the thyroid gland, and the parathyroid hormone, which is produced by the parathyroid gland. Calcitonin directly affect the osteoclast and causes bone reabsorption, while parathyroid hormone indirectly affect the osteoclast. So this is just how bone resorption occurs. Bone resorption, we have the bone matrix here. So you need to know that the osteoclasts lie in a hole shaped lacunae. Is it clear? They lie in a hole-shaped lacunae, and then we have osteoclasts. They are lacunae at the level of the periphery of the bone. So they lie in hole-shaped lacunae. You can see lacunae like that, which are hole-shaped. Just know that it is the osteoclasts. So and then they are involved in bone resorption. You see that here the bone is much more pain. It means that this one is directly going to be resorbed. Is it clear? Now this is how osteoclasts resemble. They have nuclei, like 3 lobe nuclei. So they have euchromatin and heterochromatin. You have lysosomes mostly involved here. They don't have granules, lysosomes, because they are phagocytosing on the bone matrix in order to cause the bone resorption. Now if you want to have a brief summary on the bones, just put a pause, and then you see that the bones classify into gross aspect, into compact bone and cancellous bone. Then you see the characteristics. Then you have classification based on shape. You have the long bone, short bone, flat bone, irregular bone, and then you have classification based on microscopic observation. Primary bone, secondary bone. Primary bone and secondary bone are secondary bone is further divided now into the different aspect. So from the secondary bone is further divided into the different bones that you have seen. So you can say thanks for your kind attention.